Hello, everyone. Thanks for joining us virtually this week. Hopefully, you can listen to this while you're on the road to your family or in the airport or wherever you might be. Hopefully, this will make it easy to get a little bit of this content in this week. I'm trying to make this a little bit more entertaining, perhaps, than most of the usual lectures. We're going to talk today about genotype imputation, GWAS catalogs, and what I think will be most interesting and perhaps most apt to this week of Thanksgiving is direct-to-consumer testing. So first off, I wanted to give you an overview of GWAS studies. So we talked at least a lecture about the approaches that have been used for doing genome-wide association studies. Since these studies started in about 2005, they kind of fell into a few different tiers, and so this is my general take on how the GWAS literature evolved. So from about 2005 to 2007, initial GWAS publications were released, were published. Most of these contained somewhere between 104 to maybe 4,000. That's sort of the upper limit. About 4,000 individuals. These were predominantly of European descent. And these initial studies, while a few of them did detect new association signals, most of them had relatively low power to detect new genetic variants that were associated to common diseases. So we talked on Friday of last week about complement factor H. That's a really good example of a fairly strong signal that was detected by GWAS in a relatively low number of samples. A few other notable examples, variants within the TCF7L2 gene for type 2 diabetes were discovered in a sample of around this size. So there were at least initial findings enough to convince people that doing GWAS was a good idea. So from around 2008 to 2010 or 2011, somewhere in that region, people quickly realized that you needed larger samples in order to identify these really small effect signals. A lot of people were generating data on their own, and so a lot of groups started forming to consolidate their data to analyze them together to increase their statistical power. So most of the studies in this sort of second wave era contain somewhere between 2,000 and 40,000 individuals, again, mostly of European descent. This dramatically increased power to detect these really small effect sizes. You can imagine having 40,000 people in a study gives you quite a bit more ability to detect differences in allele frequency than if you have 4,000, let's say. And then finally, from about 2011-ish onward, we started doing what I guess you would call these mega meta-analyses of really large GWAS and extending these same studies to other populations, other non-European descent populations. So most of the studies, to get a reasonable GWAS studied in this era, you needed anywhere between 10,000 and 100,000 individuals. Or, sometimes you can get away with smaller samples if you have non-European descent individuals. These studies really provided incredible power to detect really small effect sizes, really low frequency alleles. So you can identify variants that might only modulate your risk. If you had sort of a baseline risk of 1 in 10 people in the population end up getting, let's say, Parkinson's disease, then you might change that from 1 in 10 to 1.01 in 10, right? So you are very, very modestly changing your risk for any given disease by looking at these or by having these very low frequency effects. But as you sum them up, as you aggregate a bunch of them, you start to explain more and more of the risk for a particular disease. These studies also increase the allelic diversity from other populations, again by including samples of non-European descent. So you might be wondering, it's like they started combining data from multiple studies in some of these later stage GWAS collaborations. And one of the issues that comes up with that is that you might have some people, let's say I am doing a study of Parkinson's disease and I've collected all of my data on an Affymetrix gene chip array, right? So I've got around 500,000 or 1 million genetic variants all on my specific array. And my friend Jake has done a similar type of study, except he used the Illumina array because he got a better price, right? And so that means that the content of the array, the number of sites of SNPs or SNVs within the genome that I have surveyed or assayed using my genotyping array and the ones that he's surveyed using his genotyping array are different oftentimes. They vary. When you start looking at overlap, often you find that there's not a whole lot of overlap between some of these technologies. So you might be wondering, well, how can you combine data from these multiple distinct products? Well, this is where an approach called genotype imputation comes in. So basically the idea is that you can take data from different genotyping platforms with different set of SNPs and combine them. But in order to do that, you need a common reference panel that you can use where you have as much information as possible. Basically, you need a common set of samples that have been genotyped with all of these different SNPs, where you know the states of all of these various SNPs so that you know can sort of translate between one platform and another. You can sort of understand what the common information content is. So along the same time, there were two really high-profile projects which we've discussed in previous lectures, the HapMap project and the 1,000 Genomes project. So these got generated really comprehensive genetic data across multiple populations. A lot of them used trio data, so you could have haplotypes that were phased really high quality. So you could be sure that you had specific contiguous sequences of alleles on a chromosome. And most of the SNPs that were used in these genotyping arrays were actually in these projects because that's how they were discovered in the first place. This is how Affymetrix and Illumina knew that these SNPs existed and put probes on their products. And so using these data sets as a reference, as a common reference, you can build an imputation panel, a reference panel, by which you can basically infer or estimate other states in the genome. So reference panels work like this. If you're just sort of listening and you don't have the slides, you might want to look at the content of these particular slides. But the basic idea is you've got a pool of reference haplotypes that have lots and lots of allelic information. So you've got really dense genotyping, you've got sequence data, something like that. You also then have SNPs or haplotypes. Actually, it's really just SNPs. In this case, I've drawn out about 3 different SNPs that were observed in one of your particular studies, right? So from this, you can kind of look and try to build a match, right? So if you know that there are 3 alleles that you've observed in your data, 3 sets of very nearby alleles, you can try to build a match, kind of like we've done in a few other earlier examples in class, right? Build a match to other haplotypes, other stretches of sequence within this reference panel that you think are matches. So here I'm doing a little bit of color coding. You can see that you've got one contiguous blue stretch that matches very nicely, but the other strand in this observed set of genotypes might actually match a combination of different segments of reference haplotypes, right? So if you consider now, if you've got multiple genotyping platforms, you might have one set of SNPs, in this case 3, that were genotyped in Illumina, another set of SNPs that were genotyped in Affymetrix, right? And these are different sets of SNPs, but they're in the same region, they're nearby. If you remember also, due to patterns of linkage disequilibrium, right, some of these SNPs are going to be correlated to one another. So if you had the data, right, this is what the haplotypes are based on, the A's and the G's might be very tightly correlated in this data. It's just you need to know how often they appear together or they don't in order to estimate one from the other. So if you look at this, basically what you're doing here is building collections of reference haplotypes. And from a single set of data, you can project all of this stuff down by building matches. And now you know for a single individual, if you have a G at the first location, you've probably got an A at the second. If you've got an A at the third location, you've probably got a C at the fourth, and an A at the fifth, you've probably got a G at the sixth, right? So this gives you probabilities of these other alleles that you haven't really observed from your genotyping data, but that you can estimate their state based on looking at this reference set, okay? So imputation, the process of imputation is used widely in statistics. Sometimes you can infer missing data this way. So if I know, that most people's blood pressure on average is elevated as they are aged above 65, let's say, then you can sort of build a model, a mathematical model of predicting someone's blood pressure from their age alone. The same idea is being applied here, except we can do it much more precisely because we have so much better information about genetics, and we have a little bit more of a stable relationship because, again, we can rely on linkage disequilibrium and patterns of recombination rates, all of these types of information to give us really accurate models of how these alleles relate to one another. So all that said, basically what I'm saying is you can use these reference haplotypes to consolidate data sets from different platforms and build a much larger basic imputed data set. You can infer the state or the genotypes of large numbers of SNPs that surround them from the tag SNPs that they put on these arrays. So using this technology, using genotype imputation, and there's a bunch of different approaches to do this. I won't go through the details because they're quite elaborate in some cases to get these really good and high estimates of genotype imputation. Basically, what you can get out of these are oftentimes not a direct genotype call. It's very unusual that you know for certain the state of one SNP from another. Oftentimes you get a probability of a genotype, right? So if you know that there's an A at this particular location, then you can say, well, there's a 90% chance based on the data that we've got that there's a G at this second location, but there's some small amount of error associated with that. You know, there's a 10% chance that there was some recombination event and you've got a T there instead of an A, right? So sometimes we call these allelic dosages, a probability of a genotype. Sometimes you can account for those uncertainties or errors in your statistical analysis of imputed genotypes using Bayesian statistics and a few other approaches. So just be aware if you're ever dealing with imputed genetic data that there are ways to account for the uncertainty that you get out of those estimates. Okay. So to give you some sense of the types of data that are used to build these reference panels. This is a paper that was published not too long ago, 2016, where they are building, this is an ongoing effort to build a haplotype reference for large numbers of individuals, essentially sampling from global populations. So in this paper, they refer to around 40,000 total individuals, but they end up getting, actually I think they had more, but they end up with around 65,000 haplotypes. Yeah, so I think they're considering that phased, right? So that's half the number. They've got about 38,000 people, and after they QC, they drop some of them. But you can see from consolidating data across multiple different studies, in this case mostly sequencing, they were able to build these really nice reference panels. And here's a figure that's just showing sort of the increase in, as you get larger sample sizes in your reference panels, you get higher accuracy to uncover all alleles in general, but what we really look for now, like most of the common alleles are imputed fairly well. With even like the 1,000 Genomes Project, let's say, that's this orange line at the bottom if you're looking at the video. But for the most part, what we're interested in now, you often see these plots when people talk about reference panels or building new imputation approaches, because this is looking at the ability to accurately impute low frequency variance. So here, just to orient you to this plot, you've got a y-axis that's essentially the accuracy or the R squared, the aggregate R squared of predicting one SNP from another. And on the x-axis is the allele frequency. So you can see as you increase the non-reference allele frequency, you get, you know, near perfect estimation of these variants. As you have lower frequencies, you end up with lower accuracy, as you might expect. So one important component of all of this is that genetic ancestry is critical. The haplotype reference panels that are used for imputation are always going to be ancestry specific. If there's a point that I want you to take out of this entire class, it's that pretty much all of the effects that we look at are ancestry specific. So if you have a European reference population and you're trying to infer or impute genotypes in an African descent population, you're going to do a very poor job because you don't have a good reference sample for those ancestry groups. The LD patterns are different, the alleles present in those populations are different, the recombination rates are different. And so you're going to get all kinds of different, you expect to see different haplotypes in different ancestry groups. So when imputing, you really want to make sure that your haplotype reference panel reflects all of the ancestries that are available within your study. In fact, several years ago, it was a very vogue thing in statistical genetics to evaluate the differences in using reference panels with different combinations of ancestries. And the take home from all of that is that basically you want as broad a reference panel as possible. If you can include multiple ancestries in your reference panel, then do it because you'll just get generally better imputation, period. Okay, so one of the interesting aspects of GWAS is that they're very expensive to do. Lots of groups were basically generating their own data. And we now have these new approaches for combining data sets in a relatively straightforward way. You run the imputation panel, you estimate all the other markers, and now you can sort of contribute and pool your data from multiple different sample sets into one larger sample set. But in some cases, combining the logistics of combining data sets are tricky, right? So I may have enrolled studies in my Parkinson's GWAS, for instance, with a certain sort of set of understanding, with the patient population I was working with, that I would preserve their privacy, that I would never redistribute their data, that sort of thing, right? So I may have a basically like a contract or a tenant agreement with the participants of my samples, of my study, that prevent me from sharing my data, right? In some cases, my institution, right, CASE, might actually step in and be like, legally we don't think it's a good idea that you share this data for all kinds of interesting reasons, right? Just storing and transferring this data is a hassle. When you have small data sets, it's not so bad, but when you get these really large imputed data sets with millions of markers in them, just storing that securely and transferring it can be quite annoying, right? We still routinely mail hard drives worth of information around and we encrypt the data. There's all kinds of security issues that go into storing this type of data. And finally, some people just don't like sharing. There's egos, there's politics, there's, I want to be the one who finds the next Parkinson's gene and I'm not going to give my data to that asshole Jake in Miami, right? So there's that kind of thing that happens. There's like little kid books about, sharing, and when you ask adults to share and do the same thing, they often kind of balk at the same idea. But basically, this is the issue, right? Why should I share my data? I worked hard to collect this. This is my study. Why should I ever share this with somebody else? Well, so all of this stuff means that, we need to find a way, or years ago, smart people were thinking about this, about ways to share and analyze combined data sets without necessarily sharing the underlying genotypes. Now remember, there may be very good reasons why you cannot share data. If it's a protected population, a sensitive thing, if you've got, again, a data use agreement or a consent agreement with your study participants that prevents you from sharing the data, you may really want to, as an investigator, share your information and you just can't. So is there a way you can share this stuff without sharing genotypes? Well, yes, you can perform something called a meta-analysis. So if you're unfamiliar with this term, meta-analysis simply means it's an analysis of analysis results. Often meta-analyses are conducted from surveying the literature and pulling statistics out of papers, published papers. But meta-analysis is actually one way in which everybody can keep their own data, run a common analysis on all of these different data sets, and then combine their analysis results to generate a summary statistic over the full collection of data. So just like a typical GWAS, this produces a result for a single SNP. You meta-analyze by SNP. So all of the quality control issues, all of this stuff gets even more important because every study has to do exactly the same thing to give you consistent data. And ultimately, you can pool all of this stuff together and generate one overall p-value. Now, there's a couple of different ways, analytical strategies for doing this. There's a sample size based approach that's basically like a weighted p-value. There's also this approach called an inverse variance based statistic. Just be aware that there are at least a couple of different strategies for meta-analysis. I haven't seen that these really do that much of a different thing. The inverse variance approach, I think, is somewhat favored now just because in some cases that isn't as reliant. You can account for the direction of effect a little bit better. You get, better, it relies on standard error estimates and a few other things that help you run the analysis a little more smoothly. So, so now we've got a way to combine data sets, right? So we can impute them to a common reference panel. So now everybody has the same collection of genetic information. We have a meta-analysis strategy that lets everybody analyze their own data, and we can contribute it and pool it together in one single sort of collection of data. The one thing that is missing, though, is that not everybody has access to the same collection of samples, right? So you might have to really argue with someone to get access to their data, or in some cases even pay them money, right? I mean, there were really no rules about how to access data because in some cases there were logistic costs of all of this stuff. And the NIH early on in 2007 figured out that this was probably going to be a problem. So in typical NIH fashion, they come up with a solution. So they just basically said, well, if you're going to do a GWAS that is based on any kind of NIH funding, right? So if you're generating genome-wide data, then you have to deposit the data into a resource called dbGaP, which is a central repository, basically, of GWAS data sets. There are some exceptions to this. You can always be granted an exception to this rule if there are specific criteria that keep you from sharing a data set. Again, as we talked about before, then you can sort of opt out of this in ways. But otherwise, basically, this is the NIH playing the nanny and basically saying, okay, kids, everybody's got to play fair and play nice. You all have to deposit your data into this database and then make it freely available pretty much to any researcher who wants access to it. Data sets are available after an access approval. For most studies, this is relatively straightforward, but for some, they require actually a local IRB, so you may actually have to submit to your institutional review board, and we'll talk more about that later in the semester, but you might actually have to treat this study as if you were collecting it yourself. And in some cases, that can be a significant logistic hurdle. But for a lot of the data that's in dbGaP, you can just basically apply for access. It might take you a few weeks to get approval, but once you've got the approval, you can download the data and analyze it as if it's your own. So this is a really remarkable, I think, aspect of genomic science that you don't see in a lot of other disciplines. Okay. One other thing I wanted to talk about was the GWAS catalog. So this is a resource of the results from all published GWAS studies. So again, early on in this wave of generating genome-wide association data, some very smart folks at the NIH, Lucia Hindorf of most notable regard, started generating data, extracting manually the results from published GWAS papers. A lot of information from each of these studies is contained in basically this database, this catalog of SNP and trait associations. The hallmark figure of these catalogs is this thing here. So this is some kind of ideogram. I don't find these terribly useful other than to make sort of a pretty picture to put on slides. But you can note a few things here. First of all, the Y chromosome in all of its glory, it remains pristine and disease-free. Mostly because people don't look at the Y. But you can also see that there are huge numbers of trait associations scattered all across the autosomes and the X chromosome. Each of these dots is meant to represent a different sort of disease class, but you can see that there's really no pattern, rhyme, or reason to any of this stuff. Now, That said, you can go and access the data from the GWAS catalog. If you Google GWAS catalog, you can pull this up. In fact, you may have done this for your class paper assignment. But you can actually find, all of these significant associations between SNPs and traits that you might be interested in. They point you back to the original papers. You can go read their study designs, read their phenotype criteria, their classification criteria, and all of that kind of stuff. So go check it out if you haven't looked at it. It's a great way to get familiar with the type of results that you get out of GWAS studies. Okay, so for the rest of the talk here, I want to switch gears and talk about direct-to-consumer testing. So around the same time that these GWAS technologies were getting big, I think this was 2009, 2010, somewhere in there, a company came along called 23andMe. So 23andMe, again, is a completely commercial company. They partnered with a few different biotechs, most notably, I think, Illumina, to negotiate deals for genotyping costs. And their basic model for their company was this. We're going to mail you a kit. You're going to sign up, right? You pay us $100 or something around there. We will mail you a kit. You will spit into a tube about this size, which will slough off some of the buccal cells on the inside of your cheeks. You put this in, there's basically a buffer that seals when you close the cap. It's actually a pretty cool Origin kit. But anyway, you snap this thing through, a buffer goes down and stabilizes all of the cells, the DNA, this sort of thing. And you mail it back to this company. So the company then takes the DNA from your buccal cells and genotypes it on an array. Those genotypes then get loaded into an elaborate online system, and you can sort of learn things about your DNA. Okay, what can you learn? Well, when 23andMe first started, their entire business model was predicated on the idea that people would be interested in knowing and understanding their disease risks. So the initial version of 23andMe, and I think I've got some screenshots of this that I'll show you. Basically, you would log in, and it was kind of terrifying, especially if you're a hypochondriac, right? You would log in, and it would give you all of these disease risks. It's like, well, your risk of type 2 diabetes is slightly lower than the population average, or your risk for erectile dysfunction is slightly higher than, the population average, or whatever else, right? It was just this big, long list of sort of disease risk classifications. Now, granted, they did put some thought into exactly how these are displayed. So I've got an example showing here on the screen. They did try as best to do a pretty good job of explaining the average risk and then your sort of relative risk, right? So you can see here colorectal cancer, kidney disease, rheumatoid arthritis, all of this kind of stuff, right? And you can kind of see your risk compared to that of the average person. Or the average person from the population. One of the other things that they did, they got a lot of flack because GWAS associations sometimes can be, tenuous, right? They are ancestry dependent. So if you were, let's say, African American or East Asian or South Asian and you run a 23andMe disease report, they're going to base that on risk assessments from European descent populations, which may not entirely be accurate, right? For a person of a different ancestry. They also tried to weight, you can see here there's a confidence score, they tried to weight how confident they were in each of these results based on surveying the literature and that sort of thing. So this was the initial business model of 23andMe. There was a problem, however. In 2013, there was a letter sent from the Food and Drug Administration basically chastising the entire company because it wasn't, it wasn't sort of doing the type of sample tracking that was necessary for reporting clinical information. There's an entire set of standards called CLIA, Clinical Laboratory Information Assessment, or something along those lines. That basically is just quality control can you, take a sample in from, your clinic with a label on it and ensure that the results that you return to the patient correspond exactly to the information contained in that sample. You don't have sample mix-ups or something like that. So that was one issue that they had with 23andMe's sort of business pipeline. But the other thing that they really did not like was, example, your marketing, your company's markets for PGS for providing health reports on 254 diseases and conditions, including carrier status, basically as a first step in prevention and helping to take steps toward mitigating diseases, is not really kind of viable. And specifically, it then falls under a different classification by the FDA as a medical device, right? Because now you're sort of basically using this for the diagnosis, the cure, the treatment, or mitigation of disease. And so that meant that there were different standards, there were all kinds of quality things that they had to go through. So the FDA got under case. They had to extract all of the disease association information from their database and redact that back and then basically start all over. So 23andMe's been through an interesting history. We'll come back to this in a minute because I'll show you some of the data that they actually post. The other company that got on this bandwagon as well, and granted there are several others, but the other big competitor in this space of direct-to-consumer testing is Ancestry.com's DNA test, or Ancestry DNA. Again, this is the same kind of kit. It's Origene, so you spit in a tube, you seal it up, you mail it off, and you get online website access. And this is an older screenshot of Ancestry DNA, but basically you can log in here. And being a genealogy service, the predominant use of this was never for disease associations, but was really for ancestry estimation. Can you figure out one, what geographic region my ancestors might be from? And two, if I have other family members within the Ancestry DNA's genealogy database that might match, be a match for me based on my genetic makeup. Now, there's some interesting implications to all of this, but just to give you a sense, I'll go through the more modern version of this, but this is an older slide to basically give you a sense of what this looks like. This was so full disclosure, I've done both of these services. I've done 23andMe and Ancestry.com. I was given a kit for Ancestry DNA at a conference, so again, full disclosure. I purchased the 23andMe kit, I think for $50, $100, something like that, around Thanksgiving. This is when they market this stuff. And so this is kind of how I've come to know these services. You would never guess this, of course, but lo and behold, I'm of European descent. I'm mostly of Anglo-Saxon origin. All of this kind of fits with my genealogy as I know it, right? Okay, so there's a fun story I have about Ancestry.com. So I should mention that both 23andMe and Ancestry DNA have options for allowing you to connect or contact people who are genetically similar to you. When I first started this whole process, that frightened the hell out of me, right? Who's going to come out of the woodworks to, contact me because I'm their 1/16 of a cousin or something like that, right? So I completely stayed away from this. And I still, to this day, I don't think have activated any of that in 23andMe. I just sort of ignore it. For the Ancestry DNA database, though, this was a little bit different for me because there's new information I could potentially learn from this database, because they don't only, this was not a genetics company first, this was a genealogy company. And you could go online and build your family tree within their website, right? And then you could sort of search for other people who had common ancestors. You could link your family trees together. You could do all this cool stuff genealogically and discover sort of your family history. And, I've never been a big genealogy buff, but that to me was more interesting. At least then I could see how I'm related to somebody else and, what might come out of that. So there's this sort of see matches button here that I call the scary button, right? So you can click on this and you can actually see, genetically defined relatives. So when I click this, I see a screen that looks something like this, or at least I did a few years ago. Um, and most of the time you've got, a half dozen or so fourth and fifth cousins. These are pretty extended relatives. But I had one third cousin that showed up with relatively high confidence who had a really large pedigree loaded into this system, about 5,000 people. I was like, well, isn't that interesting? I don't know who the hell Susie 2101 is, but I clicked on their pedigree. She had made her family's pedigree public, so I could actually browse through the pedigree that she had posted online. And again, as I'm browsing through, again, lo and behold, there are bushes in the pedigree, right? Um, so I was like, well, isn't this interesting? That there are people here that share my last surname. This probably really is my third cousin. Just as sort of familial disclosure, um, I know the genealogy of my mother's side of the family fairly well. I didn't know a whole lot about my father's side of the family, just because my paternal grandfather was estranged from our family a little bit due to interesting circumstances. But basically, my grandmother and he divorced in the 40s or 50s or whenever. It was sort of a taboo thing back then to get divorced. But he was divorced and sort of estranged from the family. I knew him, but I didn't know a whole lot about his family. And so by matching a bush, I was actually interested to know that, hey, there's actually information here about that side of the family that I didn't know about. So, I piqued my curiosity. One of the cooler features about Ancestry is you can kind of click through individuals in these pedigrees. They show you maps because they have birth and death dates and place of birth and death. So I am from a small town called Russellville, Kentucky. You can see it right up here on the upper right. Um, and again, all of the relatives in this sort of family tree here are sort of clustering nearby geographically. And I noticed here in particular in Dover, which is not too far, maybe 150 miles, something like that from my hometown, there was this gentleman named Robert Young Bush. So I, if I go through my pedigree here, so this was my father, and he had a sister, much older sister, my Aunt Esther. Um, so my paternal grandfather was Halford. My paternal grandmother was Lou. Again, I said they were estranged. So in talking with my aunt and my father, I figured out that that my grandfather's father, so my great grandfather, was John Bush. And if I look at this guy, Robert Young Bush, he had a son named John. So I'm pretty certain that the person that I've clicked on truly is my third cousin and that we share our paternal great great grandfather, if that makes sense. So, what was really cool about the whole thing, and if there was really to be sort of a fun, happy story out of all of this, she had posted photographs of people in her family tree. So from doing this service, I actually found a photograph of my great great grandfather, whom I didn't even know who he was, right? So here it is. You can see the little black and white thing up there in the corner, right? So this is my fun story of Ancestry.com. Yeah, John Raymond Bush here. Okay, so let me show you a little bit about what these websites look like. And while we do this, I will sort of talk through some potential, pitfalls of these, right? So this is what the modern 23andMe webpage looks like. It's very different. You can see there's a bunch of different links over here on the side. They have de-emphasized their genetic health risk. Instead of showing hundreds of reports, they now show you only about 6. They are even requiring tutorials that you watch before you see the results of things like alpha antitrypsin deficiency, which can cause as a risk factor for COPD and lung elasticity, celiac disease, G6PD deficiency, which we've discussed in class, hemochromatosis, which is an HFE sort of iron-related condition, and hereditary thrombophilia, along with AMD, right? So you can click through these and see these tutorials. They give all these disclaimers, all of this kind of stuff, right? So 23andMe has actually started to focus much more on ancestry. And so if you click on their ancestry tab, you can see a report, right? I can view a report of my ancestry composition. So this is what my current 23andMe plot looks like. I'm 49.2% British and Irish. I'm 16% French and German. Broadly sort of Northwestern European, right? So I sort of come from over here. Interestingly enough, and take these with a grain of salt, these are very low percentiles and they probably have very low accuracy, but there is some West African descent and some Native American descent in my 23andMe composition at least. You can click through and see the scientific details of where they're getting their ancestry panels from, the individuals that they're pulling from, the different continental populations, and so forth. So if we go back again, I can also look at my Neanderthal ancestry. So I have 282 Neanderthal variants. This is funny, I can sort of rank myself relative to my family members on how much Neanderthal ancestry I've got, as if that's some kind of badge of honor. Thankfully, I have less back hair. Hello. I'm less likely to sneeze after eating dark chocolate, right? I mean, there's a bunch of weird crap in here. But they do give you some interesting history on Neanderthal ancestry. I have more Neanderthal variants than 61% of other 23andMe customers, but it's less than 4% of my overall DNA. So I don't have the characteristic brow ridges. Sorry, my lights are blinking over here. So this gives you a kind of sense of the 23andMe data. They also have broken out these kind of traits. These are sort of more fun things. Well, maybe back here is not so much fun, but still, they're more benign things. They're not going to get in trouble from the FDA for, showing someone their earwax type, right? So there's a few different things here that you can look through. And you can actually go through and see the genetic associations that they're basing this on, right? So am I likely to have perfect pitch or understand perfect pitch? And they even tell you in some cases sort of what the genetic makeup. Here are the actual publications that they have used to extract these genetic associations, right? And so they're basically building a model of my risk based on my own SNPs, right? So this is pretty much it for 23andMe. The one sort of nefarious thing, so you may be wondering, right, if they were marketing this thing initially as giving you information on your disease status, and now, they're basically told that they can't do that, and there's sort of this competitor out there that has much better ancestry and genealogical information. Like, how is this company still around? Well, they ended up they got a lot of people into their system in the early years of their project, right? So that means that automatically they have additional, a broader user base, so you're more likely to find someone who's related to you. They have pitched very heavily the whole idea of their genetic ancestry reports, but their true line of business comes down here under this research tab. So me as a 23andMe user can click on this thing and fill out surveys. Oh, during the last month, how would I rate my sleep quality? Well, let's just lie and say fairly bad. Have I had influenza in the past few months? I don't know. Which of the following ice cream flavors do I like best? I'm going to say none of the above. Have I had my appendix removed? I'm going to have some fun and just say yes. Let's see. Do I suffer from back pain? Let's say I just have chronic back pain. So what I'm doing here to be basically a complete asshole is I'm populating their database of research information with bogus results. Now, the reason that I'm doing this, I'm not trying to be a complete asshole, but one of the things that they don't exactly tell you is that their new business model is to share your information as a 23andMe customer with researchers precisely like me who are interested in doing human genetics research. So it's kind of annoying every now and then. I get an email in my inbox from 23andMe basically offering to sell me back the data that I've contributed to them. So you can buy access as a researcher to the 23andMe database for the purposes of conducting genetic research. But it is all reliant on users filling out questionnaires. So they have to have a sense of their own phenotype information. They have to be answering questions truthfully, which I just demonstrated is not always the case. And so there's a bunch of questions about how accurate that could all be. However, I will say, to their credit, 23andMe has published papers, peer-reviewed, and found a few things that appear to be robust findings. So the one thing I would caution you if you're thinking about doing one of these testing services, or if your family members are thinking about it over the holidays, you want to talk about this stuff, be aware that, you know, by depositing your data into these databases, they are potentially then, you know, actively trying to sell that information to other companies. That doesn't necessarily mean anything bad's going to happen to you. It just means that, you know, they're basically selling you as the product to other people. Okay, let me show you what Ancestry looks like. I haven't logged into this guy for a while, but this is what the new Ancestry DNA looks like. They've still got the basic sort of, you know, format set up here. Instead of giving Ancestry, they call this a DNA story, which I think is actually pretty cool. So again, this is my personal bias, but I think that the Ancestry information here is actually much better than what you would get out of 23andMe. So if we update my estimate, let's see. Oh, they're asking me questions. Do I expect my thing to change? Yeah, but not much. You might have increased accuracy, new regions. You might eliminate some low confidence regions. Yeah, this is about what I expected. Let's view the updated results. Aha. So they have much nicer maps. They give you kind of confidence intervals. Again, this is about the same thing that the 23andMe result gave me. Again, it matches very nicely with my own family history. We're Scots-Irish, so you're seeing, you know, a lot of influence of Great Britain. You can see that they have dramatically reduced my estimate of Central European, so I'm down to about 6%, something like that. What else is cool here, though, what I thought was nice about this is they have done a good job, I think, of trying to fit in more of like the story piece of it. So they used to have these very nice plots. I don't know if I can turn this on or not. But they basically show migration events. So there were settlers to the Tennessee and to the southern states, Alaska, Northeast Texas, from this particular region during the Irish immigration in the 1800s. And so they actually used to kind of show, let's see if I keep the update here. Aha, yeah. So here, they're actually showing these migration events, and you can kind of click here and see more about not just the DNA, but they actually give you some genealogical timelines over, you know, when these migrations happened, what was sort of going on historically, all of that kind of stuff. So this, to me, I think is taking the genetic information and spinning it in a way that better reflects, you know, the stuff that Ancestry is good at, right? So here you can actually see a lot of these migration events, how many people moved from where. So here you can see these migrations from Virginia into Kentucky, right? So from my own family history, you know, I think we were migrants from Irish, Scotland, Ireland, the UK, in around the 1750s, something like that, the 1800s, and settled, you know, in Virginia, the mountains, and sort of migrated over into Kentucky and Tennessee. So this, again, all sort of fits into my own family history and genealogy. Now that said, I didn't really learn a whole lot from doing this, right? This was stuff that I knew already. It's largely confirmatory. So that brings me to the final thought I want to leave you with on all of these discussions of ancestry. So for me, this was a confirmatory exercise. I had talked to my family members. I kind of knew our family history. Um, it reassured, these historical aspects of my own image of self. The one warning that they do not give you about any of these services is and we think about this a lot in terms of general genetic testing. If you were testing me for Alzheimer's disease or Parkinson's or something like that, there's a psychological burden that comes with knowing you have a genetic result, for some horrible disease that you can't do anything about. Some people want to know, even though there's nothing they can do about it. Some people had rather not know. Ignorance is bliss, and I kind of fall into that category. When you start talking about ancestry and these kind of entertainment style services, the one bit of psychological harm that they never make you aware of, or that they never really prepare you for, is the idea that you might discover what we call false paternity. Okay, so I have had um, two friends now who were adopted and knew they were adopted and wanted to know more information about their genetic relatives. Um, that to me makes me very uncomfortable. uh, I worked for a short time in a courthouse, and I know that at least in the state of Kentucky, adoption records are sealed and private. And there's a really good reason for that, right? So we've set up in our legal system protections for people who give up their children for adoption. And this whole system here, depending on who's using these services, could actually subvert that entire process. Because it's not just that you're, the mother or father or parents who gave you up for adoption would use the service. It's that any of their relatives would use this service. And so there's the potential that you could use this to track down a biological relative that perhaps you shouldn't contact, right? So I don't know a whole lot about the psychology of that. I'm not trying to speak to those facts. I'm just saying that there is that sort of aspect of it, that people sometimes want to use these services to understand things that, you know, without fully thinking through the consequences of those actions. The other thing that I've heard from at least two people now of happening is that they discover that their parents are perhaps, their biological parents, I should say, are perhaps not who they thought they were. Um, at least one acquaintance of mine discovered that their father was a sperm donor because their parents had fertility issues, and they figured that out that way by finding relatives in these family trees that don't match up with their own genealogical history, right? And so that prompted some very unusual discussions, and I'm sure some self-reflection and that sort of thing. You could easily imagine this happening in lots of other scenarios as well, that again, you may not even be aware of or even be thinking about when you sign up to do these services. So I guess that would be my big disclaimer if you were considering doing one of these services is, it's all fun and games until you find something really disturbing that you did not think was going to be there, right? So it's quite possible that you could find out things that might sort of shake your very understanding of who you are. And to me, that's the greatest potential harm. Who cares about, returning somebody's, probability of having male pattern baldness or something, right? The things that really bother me, I think, are that aspect of sort of shaking up, who people sort of think they are, right? Same way the same thing could happen with someone's genetic ancestry, although that's a few degrees removed, right? If I tell you that your ancestors are from a completely different part of the world than you thought before, that might actually be interesting to you, versus telling you, that your parents aren't who you thought your parents were. That's a little bit different. Yeah. The one other thing that is a little bit disturbing is that, and they've gotten much better at this, I think 23andMe initially, when they were doing these genetic health reports or reports on genetic risk, they were really sort of close to the boundary of causing problems. Because imagine for a moment if you had a family history of early onset heart attack. That might imply that you have a genetic variant in your family that's actually strongly contributing to cardiovascular disease in some way. And you might then think, oh, well, I'm going to do this online genetic test, and it says, oh, I have a low risk of heart attack, right? Um, I think the thing that was missing there, though, is that some of these genetic tests were genetic studies anyway were designed for late onset heart attack, not early onset, right? And so there may be some confusion, and you might give someone some false sense of hope or false sense of security or whatever about their own health, just because they don't fully understand exactly what all of this means, right? So I think most of those concerns are gone now that they've redacted a lot of these reports. So this is my spiel on families, ancestry, and direct-to-consumer testing. There are a few newcomers to this space. There's Nebula Genomics. This particular company has a different model where they're trying to empower you to own your own data. So the basic idea, this is kind of like cryptocurrency or something along those lines. You contribute your data in, you send them a sample, of course. They will encrypt the stuff, give you the keys to it basically, and then you sort of own your data within their system. And so you can opt in or opt out for different studies. You can even request payment or accept payments for certain studies of a particular disease. So it's a different model. They're trying to sort of disrupt this whole space by sort of giving people more privacy options and sort of founding the entire company off of the idea that they don't own your DNA or your genetic information, but that you do and that you control it. I don't know that this is really going to fly that much. I think people drastically overestimate the value of their own data in this regard. Speaking as a person who would potentially purchase information out of this system, like data like this is only useful in aggregate. So if I can buy 1 million samples worth of DNA, yeah, I might be willing to pay 25 cents a piece per genome or something like that, depending on how it works. But even that's $250,000 to access 1 million samples worth of DNA, and I don't even know if they're phenotyped in exactly the way that I need in order to run my study, right? So why not spend the $250,000 collecting people, 100 well-phenotyped people in the clinic and be able to do whatever I want to with them? So it's not entirely clear to me that this whole sort of model is going to work. And I think most people, the thought of paying $100 to put my DNA in a system where I'm only going to get a quarter or $1 per access or something like that, I think is going to turn most people off. I don't think that's very well thought out yet. So that's all I have for the lecture. I hope you enjoyed this. I hope it was at least something you could listen to. Hopefully it'll spark some fun conversations with your family members over Thanksgiving. You can talk about 23andMe, genetic testing. You can figure out where your own genomes were inherited from across the globe. Thank you so much for tuning in, and I will see you. I actually will send an announcement. I am out of town next week. I am being drug away to a conference in Paris. I'm so terribly sorry. I will eat lots of cheese and drink lots of wine for you. The lecture on Wednesday will be given by Dr. Dana Crawford. She'll continue our odyssey into the fundamentals of genome-wide association studies. Dana's done tons of GWAS studies herself, including many on non-European descent populations. She actually had a result in the 23andMe database for hypothyroidism. You should ask her about it. It's kind of a fun story. And then on Friday of next week, Jessica Cook-Bailey will present on the ethics of conducting genetic studies, which will follow up on some of the interesting issues I think we touched on today. But Jessica's awesome at thinking through these problems, and I think you'll have a fun lecture with that. We will convene again the first week of December. I will see you again then, at least. So I hope you have a good Thanksgiving, a good holiday, and